Well, good morning, everyone. My name is Jonathan E Zul Luna, and I'm a fourth-year computer science student at UPI. These here are Eastern oysters, and for Prince Edward Island, they're much more than just seafood. They are an essential part of our economy and our coastal identity. However, this industry is facing a serious threat from a microscopic parasite that causes a disease called MSX. Today, I will talk about my summer research and my current honors project, where I'm focusing on finding a way to help experts diagnose this disease much faster. But before we talk about the technology behind this project, I want to give you an introductory context and just talk about why this work is so important for our region. So as I mentioned before, in PEI, the oyster industry is basically woven into our identity. Financially speaking, it is a huge economic driver for the province, bringing in over $24 million every year. Additionally, it supports the jobs of hundreds of people in the rural and coastal communities that define the island. So we have this very important industry, but as we know, it faces a serious, persistent threat. That threat is microscopic. So the enemy is extremely small, meaning that we can't just look at an entire oyster on half the shell. We have to look at inside the tissue itself. And the tool that allows us to do that is called a histology slide. Here you can see exactly how we get from a whole oyster to the digital image that we analyze. On the left, you can see the standard oyster. The yellow band there highlights the very specific region where a very thin cross-section of the entire body is carefully cut. After a multi-day process of preservation, staining, and scanning, that single thin slice of tissue becomes the high-resolution image you see here on the right. This is what we call a whole slide image. Now that we understand what a histology slide is, let's take a look at some examples to see the threat we're trying to find. The threat is a parasite called Haplosporidium nelsoni, which causes MSX disease. The best way to understand the damage it does is simply to see it. Here we have tissue from a healthy oyster. Notice how clean and structured it looks. Well, now compare that to the tissue on the right. This is from an oyster with an advanced MSX infection. Even if you're not an expert, you can see a clear difference. The tissue on the right is a bit more chaotic. It looks more purple. It has this stronger purple coloration, and the normal structure is breaking down. That is the result from the parasite literally destroying the oyster from the inside. But now let's take a look closer to see exactly what's going on in more detail. This here, at this magnification, we can see the direct impacts. The arrows there point to the parasites in its spore-releasing stage. But do you see all those small, dark purple dots surrounding that area? Well, that's the oyster's immune system trying to swarm the parasite to destroy it. That's what was causing that purple coloration in the previous slides. So this is an intense inflammation response that aims to destroy the parasite. However, it destroys the oyster tissue in the process, which ends up killing the oyster. And since there's no cure, our best defense is early and accurate detection before that disease spreads to other oysters. So how do we detect it? That all happens in the Atlantic Veterinary College, or AVC. They are the main histology-based diagnosis center in Atlantic Canada. There are other diagnosis methods, like previously talked in other talks, like PCR, which can detect the parasite's DNA. And this is great for a binary answer of yes or no, but if we want to understand the severity of the disease in a sample, we need to go with histology. That means we need a trained expert that can look at the tissue under a microscope. However, the challenge here is that this process is entirely manual. So it relies on a very small number of pathologists. Something there. In our case, the AVC has a single pathologist, Dr. David Groman, and he's looking at thousands of slides for the entire Atlantic Canada region. He mentioned that he has received over 6,000 samples just in the period from April until today. It's just not possible for him to go through all these slides in a short period of time. So what we're trying to do is help him build a custom tool that can assist him in this process. And in any machine learning project, our first step is to analyze our data. However, we have no data. There's no publicly available MSX dataset, which means that we need to ask our pathologists that are very busy to spend countless of hours analyzing through these slides and outlining every single of these parasites, which is kind of contradictory. We have a paradox. We want to build a tool that helps them save time, but to build that tool, we have to ask them to spend hundreds of hours. So our goal, our first goal, is focused on breaking out of this cycle. Our strategy is called a human-in-the-loop approach. Instead of building an AI in isolation, we create a cycle between the expert and the machine. So first, we have this first pass where we perform a pre-analysis on an Oyster slide, and we generate an initial set of annotations with flags for any potential areas of interest for our experts. Next, the expert reviews all these annotations, and this is where the time saving happens, because instead of looking at the entire blank slide, they just have to look at the annotations given by the AI, and they just correct them and validate them. Then we use this data to retrain our AI, which means that with every new slide we process, our AI becomes smarter. So to make that human-in-the-loop strategy a reality, I created a complete automated pipeline. However, we found a critical challenge along the way. Before we can even look at the parasite, we have to solve a much more basic problem, figuring out which tissue belongs to which oyster. And you may be wondering what I mean by this. Well, most of the oyster slides in the ABC contain more than a single oyster, and this is done because it cuts costs and it improves efficiency. However, this is what creates our challenge. Sometimes you get a perfect slide, just like this. The sections are very well defined and they're very clean. So writing code to separate both of these is fairly easy. However, we often encounter something much, much messier. Because the slides are prepared by hand, as we previously saw, they are often fragmented, so they're missing pieces, they're overlapping or even touching. That simple approach we could use for the perfect case would not work at all in this one. So we needed to first build a robust approach that was able to outline each oyster separately, ensuring that we could perform well in the following steps. So we have that initial step. We need to teach our computer how to accurately find the oysters on the slide. And to find the best technology for this job, we didn't just use one method. We tested many different AI approaches to see exactly which one would be the most accurate. But to compare them, we first needed a clear way to measure their success. And that's where we used the DICE score, which basically measures the overlap between the expert's annotation and our model's prediction. So if they match perfectly, we get a DICE score of 1. If they overlap pretty good, they get a higher score, and if it's a poor match, the score is pretty low. So basically, the higher the score, the better. Here we have the detailed results from our investigation. This table shows the performance for each of the methods we tried, but to make the key takeaway easier to see, let's visualize this as a simple bar chart. So this chart makes the story a little bit clearer. We started with a classical algorithm that was our baseline, and we achieved a pretty good score of around 0.75, but it was not enough to be considered reliable. So then we moved to a standard AI model, which is what is usually used for other histopathology AI projects. However, we achieved a very low score, which likely comes from the amount of data we have, which we will address in the future slides. Lastly, we have our hybrid approach, which is a massive improvement compared to the other ones. We achieved an accuracy score of nearly 0.9, proving that this was the most robust and data efficient method that we could use. So we're confident that we found the right technology for the job. So let me briefly show you how this winning approach works. Our first step answers a simple question. Where is the tissue? I wrote a script that quickly scans a lower resolution version of the slide and then looks for the general location of the oysters, and it generates a set of simple prompts or instructions for the next step. In step 2, we pass those instructions to a powerful foundation model called Segment Anything Model provided by Meta AI. This model aims to answer a much harder question. What are the exact boundaries for the oysters? So we're basically telling it, the thing I want you to find is inside this box. It includes the green dot, and it definitely doesn't include the red dots. So since Sam has seen billions and billions of images, it is really good at following these simple instructions and producing a precise outline. So here we have a direct comparison where we can see in blue the expert's ground truth, their annotation, and in green is the guess made by our model. So we were able to solve that segmentation issue. However, our analysis gives us a very clear diagnosis of the system's two main limitations. The first and most significant is that this first part I talk about is brittle. So in really, really hard slides, it still has some issues. Here we can see a progression where in the intermediate steps, it merges the oysters, so that just results in inaccurate masks at the end. And then on the broader spectrum, I talked about how we don't have a lot of data. Well, what I meant by that is that we have right now 30 available slides. We can get many more, but those are very large. And what this implicates is that we are not able to capture the natural variability that you would encounter in the actual field. So this points us to a clear guide on what we will do next. And that starts with our two-phase plan, where first we will use this human-in-the-loop system, which is successfully built, to get more slides and train this loop I mentioned before to make the system better and better and generate that data set that we were missing. And once we have access to that extended data set, we will develop a field-ready assistant tool that will help the pathologist process these slides much, much faster, and it will be validated and trusted, and we will ensure that it is explainable so that they can trust what the AI model is saying. So to wrap up, in the summer, we took this project from an idea to a fully engineered pipeline. We solved that segmentation challenge I mentioned before with a data-driven approach. And lastly, we have turned our results into a clear plan for what comes next. At the end of the day, this work lays the foundation for a tool that can protect one of the most important industries on our island. Thank you. I'll be happy. Bueno.